Okay, welcome to the course of Intelligence, Healthcare and its Applications, and this is the part 2 of the cluster analysis and its applications. So in today's course, we will not do the workshop because I think there are many important concepts behind the cluster analysis, and also I want to show you some real applications, and then we will do some workshop in the next week. Okay, so today I want to show you a very interesting applications that I have developed quite a while ago. It's almost about the 18 years ago. So you know that the cluster analysis is not really a new machine learning methods. The idea of cluster analysis is already developed for many decades, and the most important thing is that how you define the distance in this cluster analysis. So in the previous part, I already showed you that you can use the Euclidean distance, you can use correlations, but there may be other distance measures that can help you define the distance between different types of data. So in the biology or in the physiology, we really have different types of the information. For example, in the nervous system, then we will measure the neuronal impulse as the informations, and of course we have a genetic codes as our informations. And then if we do the electrocardiogram recordings, then we will measure the human heartbeats, and that is also one of the important informations in our cardiovascular systems. So the others informations are very intuitive. For example, if you look into the earliest records of the painting by human, then probably you will see this is kind of the horse, and that you can still easily identify the source of information just by looking at this painting. And also, the human also develop a very sophisticated writing systems, and this is cuneiform scripts by Sumerians in 2600 BC. Then you can see that there are different types of the sculptures, and then to define the different types of words. So the question is that how to effectively categorize information of different origins. So for example, how do we classify human genome and chimpanzee genome? And that is the question. And a general principle to analyze the information embedded signals is the repetitive patterns. So for example, in the case of the writing system in the literary text, the repetitive patterns is our words, right? So for example, if you look into the plays by the William Shakespeare's, and here I just show you two plays. The one is the Winter's Tale, the other is the Cymbeline. Then you can calculate the frequency or the occurrences of each word here. So for example, in the play The Winter's Tale, the most frequent words here is the word the, and it has 857 occurrences in the place The Winter's Tale. And if we find out all frequencies for each words, then we can arrange these words according to their rank. So for example, we can place the first rank according to the most frequent words in the display, and the word I is the second frequent words in display, and etc. Okay, so likewise you can also do the same thing, do the same statistics in another play. In this case, in the play Cymbeline, we also find out that the most frequent word is the word the, and the second word is the I, and the next one is and the next one is to. So if you try to analyze these frequency and rank order statistics, you'll find out that, oh, they are quite similar between two different plays. So these are two plays by the Shakespeare's. They talk different stories and they have different characters, but if you look at the frequency and rank order statistics of these most commonly used words, the rank of these words are quite similar. So if you compare the plays from different authors, you will find out there actually a difference between these frequency rank order statistics. Okay, so this is a play by John Fletcher. This is a play Banduka. So in this case, the most frequent word is the word and the second one is the, and the third one is I. You can see that the rank of these words is quite different from those used observed in the Winter's Tale, which is by the William Shakespeare. So it gives us an intuition that maybe there are these frequency and rank order statistics are the features of the style of the authors. So probably from the previous course, you will think that, okay, we can measure the correlations, we can measure the distance. So in this case, I will use a special visualization way to measure the distance between these two rank informations. So for example, if we have the rank information of each word from one play and also in another play, then we will just use this rank information to plot this on a two-dimensional plot. And so the horizontal axis is the rank from the one play, and the vertical axis is the rank from another play. So in this case, the word the has the first rank in the Winter's Tale and the third rank in the Banduka. So I will plot the word the here, and likewise we can plot the other words according to its rank information. So if in this way, then we can simplify the multiple parameters only into one parameter, which is the rank. So if the two plays have the same word rank order statistics, you will expect these words will be placed on the diagonal line, which means that they have the same rank order statistics. But in this case, you will observe that they have a quite different rank order statistics because the words are away from the diagonal line. So let's compare the results from the comparison between the same author, which is the Sibling and the Winter's Tale, and the case between the two different authors. So in this case, it's Winter's Tale and the Banduka. So you can see that in the case of the same author, the rank order statistics are quite similar. But in the case of the different authors, then these rank order statistics will be very different. So it's even true in the Chinese language. So if you compare the martial art fictions by Jin Yong and also by Gu Long, then you will find out that the distance between Jin Yong are very similar. But when you compare the rank order statistics of the fictions from Jin Yong and Gu Long, you will find out that distance are quite large. Okay, so I defined a distance measure that I called the information-based similarity index. Then I published this paper in 2003 in the Physical Review Letters, which is a very top journal of the physics. Also a very old journal of the physics, and it has a very profound applications. And so once we have the distance measures, then we can apply the cluster analysis to this distance data. So for example, if we have a sibling and winter's tail and a Bonduca, then we can use the rank order statistics to measure the distance between the sibling and the winter's tail. Then we can also measure the distance between the sibling and Bonduca, and also the Bonduca and the winter's tail. Then we can define a triangular relationship between these three places. Then of course you will see that the distance between the sibling and winter's tail is smaller than that between the Bonduca. So using this relationship, then we can use cluster analysis, which is a hierarchical cluster analysis, to define the cluster, which is the sibling and the winter's tail, and the Bonduca is away from this cluster. Okay. Okay, so this is the kind of validation of this method. So here we have 16 different texts or the books from the different authors. So for example, we have the detection fictions from Conan Doyle. Here the values of fears, the science of four, and also we have fictions from the Jack London, the Sea Wolf and the Call of Wild, and also we have the Alice Adventure in Wonderland, and so these are very famous fictions or texts. And so using the rank order statistics and information-based similarity index, then also we apply the cluster analysis based on that distance measure. Then we can correctly identify the authorship in based on this distance measure. So I think this result can give you more intuitive idea behind the cluster analysis, because once you have a right distance measure, then you can actually find out the very nice classification of the data. Okay, so here I show you one of the very beautiful application of these methods, which is to solve the authorship debates in the Chinese literature. So there's a fiction called, it's not actually fiction, it's a kind of a Chinese novel, which is called The Dreams of the Red Chamber, and in Zhong is called 红楼梦. Then the story behind the Dream of the Red Chamber is that not only it is one of the China's 4 great classical novels, and also it was appeared to have 80 chapters in the first appearance. So it is written by the Cao Xueqin in the middle of the 18th centuries during the early Qing dynasty. And originally it has only 80 chapters in the first print. Then later on, and there are 2 scholars claim that they found the other 40 additional chapters in the market, and they try to do some editings on these 40 chapters, and they then claim that they complete the novel. So Gao E and Chen Weiyuan actually they claim that they found out the 40 chapters by the Cao Xueqin, and they try to do some editing. However, many people believe that these additional 40 chapters are not originally by Cao Xueqin, because there are many essential difference between the first 80 and later 40, and there's a very famous scholar called Hu Shi, and he actually wrote a book trying to use some scientific evidence to to show the difference between the first 80 and later 40 chapters. And also I want to show you some interesting findings, because there's also one scholar in Taiwan. He has a very interesting study that he tried to calculate the number of the different vegetables or plant. Then he found that in the first 80 chapters, in average, there are more than 10 vegetables or plants appeared in one chapters in average. But in the later 40 chapters, there may be only 3 or 4 vegetable or plants. So it shows maybe that the abundance or knowledge about the plant or vegetables by different authors. So people believe that the later 40 are by Gao E and Chen Weiyuan, not originally by Cao Xueqin. So if we look at the rank order statistics of the words used in this text, then in this case, we divide the data into the first 40 chapters and the middle 40 chapters and later 40 chapters. And you can see that the rank order is quite similar between the first 40 and the middle 40 chapters. The rank order are very similar, but it's quite different in the later 40 chapters. So immediately you can see that the similarity is less is similar between the later 40 compared to the first and middle 40. So if we divide the all 120 chapters into group by the 10 chapters, so this is the first 10 chapters, the second 10 chapters, and until the 12 10 chapters. Then I calculate the pairwise the distance between these 10 chapters, then do the cluster analysis. So this color represent the distance between 2 10 chapters comparisons. So for example, here you can see this is comparison between the first 10 chapters and the 71 to 80 chapters. Okay, so the red color means that there are larger distance, and the yellow color means that there's a smaller distance. So we can classify this cluster this data, the distance matrix, into the clustering tree. Then you can find out that okay, the later 40 chapters has a very large distance compared to the first 80 chapters. So that is the one compelling evidence that the first 80 is different from the later 40. And even the first 20 chapters are quite different, because if you look into the Hu Shi's books about the dream of red chamber, he has actually calculated the number of the word change in the first 20 chapters is also very significant. So that is one of the reasons that you can find out actually the first 80 and the later 40 are quite different. And also there are significant editings in the first 20 chapters. Okay, so I also applied this concept to the comparison between Shakespeare and the Fletcher. So you know, in the older time, there's the author. There's also collaboration between authors. So in the modern time, we wrote papers by different authors. So likewise, in the older time, these playwright, they also wrote plays together. So for example, Shakespeare and Fletcher wrote the two novels, Kingsman and also the Henry VIII together. And we know that in the case of Henry VIII, they are more influenced by the Shakespeare, and also in the case of two novel Kingsman, there's more influence by the Fletcher. So in this case, we compare the result of cluster analysis also include the original play by the Fletcher and also the plays purely from the Shakespeare, then you can see that the tree actually reflect the intermediate relationship between the Fletchers and the Shakespeare. So the Two Noble Kingsmen is closer to the Fletcher because he has more written by the Fletcher, and Henry VIII is more closer to the Shakespeare because it's written more by the Shakespeare. And also we can use this method to identify the chronology of this play. So if we assign the root of this cluster or phylogenetic tree, then we can roughly identify the early place, the middle place, and the late place by the Shakespeare. So this tree is actually meaningful even in the time. And also in the Marlowe's place, we can also identify the chronology of the Marlowe's place. And also we find out that there's a play called Edward III. It was published anonymously in the first print, and nobody knows who wrote this play. But many people argue that this is written by the Shakespeare, and the others arguing that this is written by the Marlowe. And we found that this, according to the chronology, it should be placed in the Marlowe's play, not in the Shakespeare's play. So that is one of the beautiful results that we can use the distance measure to find out the relationship between different text. And also, of course, this method can be used in the medical applications. So in this course is about the medical the applications of machine learning. So in this case, we apply this method to classification of the human heartbeat. Then we know that the human heartbeat is regulated by the autonomic systems. And to simplify the representation, that we know that the heart rate will be increased by the sympathetic stimulation, and the heart rate will be decreased by the innervation of the parasympathetic stimulations. So our heart rate is not a constant number. It actually goes up and down according to regulation of autonomic systems. And the one way to feel about this is try to breathe very deeply. So you can try to do this. You try to inhale for like 5 seconds and exhale for maybe the 6 to 8 seconds. And you will feel drops in the heart rate during the exhalation. And that is because of the regulation of the autonomic system to prevent you from to keep the constant cardiac output. So you have increased heart rate during the inhalation because expansion of the lung will compress the veins and returns to the heart. So that the heart rate need to goes up to compensate the reduced the input of the blood. And in the contrast, the heart rate needs to goes down because during the exhalation, the blood is increasingly returned to the heart. Then the heart rate needs to goes down to compensate the increased preload. So that is the heart rate dynamics essentially. And the question is that can we classify the heart rate into different physiological conditions just based on the different types of the heart rate fluctuations? And in this case, the heart rate, there's only one healthy heart rate in this case. So the first one is heart failure. The second one is also heart failure. And this one is atrial fibrillation. It's very irregular. And only this one is from the healthy person. So using, so how about we can use the same technique I illustrate in the literatures to classify the heart rate into different clusters. So to do this, we need to map a heart rate into a symbolic sequence. For example, we cannot just use the raw heart rate data because there are too many patterns. How about we just simplify these patterns into a symbolic sequence? Means that can we translate these heart rate patterns into a kind of symbolic representations? And in that case, we need to define the words of the heart rate symbolic sequences. So the way I do this is very simple. I just calculate and try to identify the up and down patterns of this heart rate sequence. So in the case, so this horizontal axis is the bit number, and the vertical axis is the interbit interval. And in the case of the slowing heart rate, we can use this as, and then assign this as a number 1. And in the case of the increasing heart rate, we can assign this as a number 0. So we can transform these up and down patterns of the heart rate into a sequence of the 1 and 0. So in this case, we can transform this heart rate into 11000 and 11001100. So it's kind of binary sequence. Then we can further define the word of this sequence using an n-bit word. So in this case, we use the 8-bit word, which means that we first capture the first 8 sequence, which is 11000110, the first 8 sequence. And then we'll use the sliding windows and move to the next 8-bit, which is 10001100. And similarly, we can move the window for the whole binary sequence to generate different patterns. And this binary number is actually corresponding to a decimal number, right? So for the 8-bit word, we have 256 combinations, which is equals to power of 2 in 8. So so different binary numbers can be translated into different decimal numbers. So in this case, we can calculate the frequency or the occurrences of these patterns in these heart rate sequences. And if there are similar patterns, then you should observe like this one. So this is the comparison of the heart rate patterns between 2 young healthy subjects. And if you compare the healthy and the patients with chronic heart failure, then the heart rate patterns are very different. So that is using the same technique, we can easily identify the physiological conditions and to make a cluster analysis of this heart rate of different conditions. So in that study, I use a cluster analysis to make an inference between a healthy young person, healthy elderly participants, and also patients with congestive heart failure, and also atrial fibrillation. So you can see that the atrial fibrillation is very close to the white noise. It means that this heart rate pattern is very irregular. And also the healthy elderly and healthy young are clustered together, and they are different from the patient with congestive heart failure. Okay, so probably you can think that this method is very universal, so we can apply this virtually to any type of the data. So in this paper, we try to cluster different healthy subjects according to their heart rate patterns, and we found that in these 2 clusters, they have different genotype regarding the beta AR gene, which is a gene related to the heart rate regulations. So, so that is one example that we can use a cluster analysis of the human heartbeat. Not only genetic profiles, but also the human heartbeat is quite a time series data. Then we can cluster this time series data into different clusters. And of course, we can apply this to the genetic sequences. And it's very similar between the genetic sequences and also the literature data. So we use different words as a sequence. And likewise, in the genetic data, we have different codons or genetic the base pairs. So for example, for the DNA, the words is the kind of a codon. So we can use the 3 tuple base pairs to define the word. Then we can also find out the frequency of this sequence in a particular words. For example, here, the word CCC is most frequent patterns in this genetic sequence, and likewise in the other patterns. So in a comparison of the mitochondrial DNA, and I should emphasize that mitochondrial is from mother. So mitochondrial DNA is kind of a very good indicator of evolution, because the mitochondrial DNA will be vary from the species to species. So if you compare the mitochondrial DNA between the same species, which is the 2 human, you have a very similar the rank order statistics. But in the case of the comparison between gorilla and the human, if you just line these 2 sequences to find out difference, actually they are still very similar. But in our methods, we can find out there are significant difference between the genetic sequence between the gorilla and the human, based on the rank order statistics. And even we can use this method to trace the evolution patterns of the viral genome. So the influenza genome is one of the virus that has a very high rate of the mutation. Then you can see that we have the earliest sample we can trace is 1930, the strand of the influenza virus. And later on, there are several outbreaks in either in United States or in Asia, and also in USSR, which is the previous country of the Russia Federation. So using the cluster analysis, and also we translate this into evolutionary relationships, then you can find out that there are 2 evolutionary lines of these different types of the viruses. So for example, this strain can be traced to the 1901, which is a Spanish outbreak, and the other strain can be traced to the USSR, the strain, which is very consistent with the paper published in the 1986 Science that is based on the alignment technique. So this distance measure is very useful in identifying the origin of the data. So for example, in the previous, we published the origin of the SARS virus, coronavirus, using this technique, and we compare the segments of the SARS genome to different groups of the coronavirus, and find out that the coronavirus, particularly the SARS coronavirus, is very heterogeneous. It has a combination from the different groups of the coronavirus, and it's a very dirty genome. And it is also supporting the idea that the SARS coronavirus has underwent a very high rate of the mutation. So this is also true in the current pandemics, which is caused by the so-called SARS-CoV-2. And we know that SARS-CoV-2 is very similar to the original SARS. So just like in our published data that we use our method to compare the SARS-CoV-2 to the other coronavirus, we found that this SARS-CoV-2 is very close to the SARS virus and also the SARS-related bat virus. Okay, so this is one of the good applications of you can use a distance measure to find out the relationship of different viruses. So this is, I hope you can give a very general idea of the cluster analysis, that this cluster analysis can be applied to many types of the data. So you need to choose the right variables and right distance measures, then you will find out the new data structures of this raw data. So there is no workshop in today's course, but I hope you can find out more resources on my MATLAB toolbox. So you can look for the information-based similarity toolbox on the internet, and I also give you a web link here. Then you can download the MATLAB toolbox, and you will find out a very clear tutorial I written for this toolbox. So I hope you enjoy this toolbox, and you can use this toolbox to do all analysis I show you today. Okay, thank you very much, and so I hope you enjoy this course, and we will continue our this cluster analysis application in Alzheimer's data in the next week. Okay, thank you very much. Bye-bye.